Ever wondered how our brains age and why Alzheimer's disease occurs? In the intricate dance of cellular biology, where every molecule plays a critical role, recent findings have spotlighted a rather enigmatic player, glutamate, tRNA fragments. This discovery stems from groundbreaking research led by Professor Liu Qiang at the University of Science and Technology of China. His team's study, published in the esteemed journal Cell Metabolism, dives deep into the molecular mechanisms that underscore brain aging and the progression of Alzheimer's disease. Brain aging is a natural, albeit complex, process marked by a gradual decline in cognitive functions. Alzheimer's disease, the most prevalent form of dementia among the elderly, further accelerates this decline. The crux of Professor Liu's research hinges on a particular type of transfer-RNA-derived small RNA known as Glu5 tsRNA-CTC. This fragment accumulates with age in the mitochondria of glutaminergic neurons, disrupting crucial cellular processes. By impairing mitochondrial protein translation and degrading the structure of mitochondrial cristae, Glu5 tsRNA-CTC plays a pivotal role in hastening brain aging and the pathological cascade of Alzheimer's disease. This discovery could reshape our understanding of the aging brain and Alzheimer's disease. What exactly are glutamate tRNA fragments and why are they important? Let's delve into the enigmatic world of these molecular fragments to understand their pivotal role in brain health. Glutamate tRNA fragments, or Glu5 tsRNA-CTC, are small RNA molecules derived from the transfer RNA for glutamate, one of the most abundant neurotransmitters in the brain. In a healthy brain, these fragments play a critical role in maintaining mitochondrial function. Mitochondria, often referred to as the powerhouses of cells, are crucial for energy production and overall cellular health. However, as the brain ages, the scenario begins to shift. Research has demonstrated that there is an age-dependent accumulation of these glutamate tRNA fragments in the mitochondria of glutaminergic neurons, neurons that primarily use glutamate as a neurotransmitter. This accumulation is not just a harmless buildup, it has profound implications for brain function. The excessive presence of Glu5 tsRNA-CTC in mitochondria disrupts the normal protein translation process essential for maintaining mitochondrial structure and function. This disruption is particularly detrimental to the mitochondrial cristae, the folds within mitochondria where energy production occurs. As the structure of these cristae deteriorates, the neuron's ability to produce energy efficiently is compromised. Furthermore, this accumulation impairs the brain's glutamine metabolism and synaptic structure, pivotal for cognitive functions such as learning and memory. It sets the stage for accelerated pathological processes associated with brain aging and, more critically, Alzheimer's disease. The accumulation of these fragments disrupts essential brain functions, a key factor in cognitive decline. How do these fragments interfere with our brain's powerhouses? Mitochondria, often referred to as the powerhouses of our cells, play a crucial role in brain function by providing the energy necessary for neuronal activity. In the context of brain aging and Alzheimer's disease, mitochondrial health is paramount. The study led by Professor Liu Qiang at the University of Science and Technology of China has shed light on a specific molecular saboteur within this energy-generating system, Glu5 tsRNA-CTC. This fragment, a transfer-RNA-derived small RNA, accumulates in the mitochondria of glutaminergic neurons as we age. Normally, these mitochondria are sites of robust protein synthesis, critical for maintaining the structure and function of the cell. However, Glu5 tsRNA-CTC disrupts this process by impairing the binding and function of tRNAGlu, a key component in the mitochondrial protein translation machinery. As a result of this impairment, the aminoacylation of tRNAGlu is hindered, which is a vital step for the incorporation of leucine into new proteins. This disruption leads to a cascade of effects. Mitochondrial-encoded proteins are not synthesized properly, which in turn affects the mitochondrial cristae structure. These cristae are folds within the inner membrane of mitochondria that are essential for optimal energy production. The consequences of such disruptions are profound. Impaired mitochondrial function means less energy for brain cells, leading to reduced synaptic activity and decreased levels of synaptic glutamate. This decline in glutamate levels is particularly detrimental because glutamate is a major neurotransmitter involved in learning and memory. Impaired mitochondria lead to significant setbacks in brain function, accelerating aging and Alzheimer's pathology. Can we reverse or mitigate the effects of these tRNA fragments? This question is at the heart of groundbreaking research led by Professor Liu Qiang and his team. They have embarked on an innovative journey to understand and potentially counteract the adverse effects of Glu5 tsRNA-CTC in brain aging and Alzheimer's disease. The approach utilized by Professor Liu's team involves the use of Antisense oligonucleotides, a type of genetic intervention. These molecules are designed to specifically target and bind to the Glu5 tsRNA-CTC fragments. By doing so, they effectively reduce the levels of these fragments within the neurons. The mechanism behind this is fascinating. Antisense oligonucleotides are short strands of DNA or RNA that are complementary to a specific mRNA or tsRNA sequence. When they bind to their target, they block the biological processes that would normally be triggered by the target RNA. In this case, they prevent the Glu5 tsRNA-CTC fragments from impairing mitochondrial function. The results of this intervention have been promising. In laboratory tests, aged mice that received injections of these antisense oligonucleotides directly into their brains showed significant improvements. Most notably, there were measurable enhancements in learning and memory capabilities. This suggests that reducing the levels of Glu5 tsRNA-CTC can protect against or even reverse some aspects of cognitive decline associated with aging and Alzheimer's disease. Moreover, this approach has broader implications. It highlights the potential for RNA-based therapies in treating neurodegenerative diseases. By targeting specific dysfunctional RNA molecules, researchers can potentially restore normal cellular function and prevent the cascade of events leading to neuronal damage. This breakthrough offers a hopeful avenue for potentially delaying or reversing age-related cognitive decline. As research progresses, the hope is to refine these therapies, making them more effective and accessible, paving the way for a future where the mysteries of brain aging are not just understood, but also effectively managed. What does this mean for the future of treating brain aging and Alzheimer's? As we have explored, the discovery of glutamate tRNA fragments' role in brain aging and Alzheimer's disease marks a significant stride in understanding these complex conditions. The accumulation of Glu5 tsRNA-CTC in the mitochondria of glutaminergic neurons disrupts crucial processes, accelerating the onset of cognitive decline and Alzheimer's pathology. By targeting these tRNA fragments, researchers have opened a new avenue for potentially mitigating the effects of brain aging. The successful use of Antisense oligonucleotides in aged mice, which alleviated learning and memory deficits, is particularly promising. This approach not only highlights the therapeutic potential, but also underscores the importance of maintaining mitochondrial health for cognitive function. However, the journey is far from over. The intricacies of mitochondrial dynamics and their impact on brain function necessitate further research. Understanding the full scope of tRNA fragments' roles and developing targeted therapies will require continued innovation and collaboration in the scientific community. With ongoing research, the future could hold new ways to protect our brains from the ravages of time and disease,